This is Chapter 8, Microbial Genetics. So this is the plasmid DNA from E. coli, and so what they've done is they've lysed the cell membranes and the cell walls, and they've released and stained the DNA from from these cells. And so what you're seeing here is just the circular DNA that these bacteria have. So the big picture here is genetics is the science of heredity. And the central dogma of modern biology is essential, and it's the key to understanding all of this. We can also have mutations, and we can have gene expression is going to be controlled by operons. And so typically we've always talked about the central dogma of modern biology, and we talked about it in A&P, we talk about it in biology, and so what we do is we say the DNA gets transcribed into RNA, which is then translated into proteins. And so those proteins have some type of function. Now I'll also draw a circle here. And so this is synthesis, so DNA synthesis, and we'll talk about that as well, because it's a very important part of the cell cycle, and it's one of the things that actually makes the cell cycle useful. So we can copy our DNA. It's a very easy thing for our cells as well as the bacterial cells to copy. Now, mutations can alter a genome, and so you can take mutated DNA, which is going to make mutated mRNA, and it's going to have an altered protein and an altered function. And so it's going to go through the same steps. And so here we have a couple of different mutations. And so we can have a base substitution mutation where we go from a C and a G right here in this third position to an A and a T. We can also have a frame shift where the DNA base pairs are added or removed from the sequence, causing a shift in the sequence when it actually reads the sequence and converts it to amino acids. So an inducible operon includes the genes that are in the off mode with the repressor bound to the DNA, and is turned on by the environmental inducer. And so this inducer right here comes in here, and it causes it to get off of the DNA essentially. A repressible operon is the opposite. So the DNA is generally in the on position, but it's turned off by the repressible operon. So the alteration of bacterial genes and gene expression is one of the causes of disease, and it can also prevent disease treatment, and it can also be manipulated for human benefit. And so this is something that's a very fast-growing kind of thing that we're seeing a lot of now. So here we can take a look at a couple of examples of how mutations are helping us and also hurting us. And so the diseases here, they can start to create proteins that are toxic to humans as well as to animals and other organisms. We can also get antibiotic resistance, and that's where we have these bacteria that are becoming resistant, and there's a list of them. And these are getting to the point where it's becoming a crisis, where if we don't figure out how to treat these, then we're going to lose the effectiveness of the drugs. And then lastly, down at the bottom, bacteria have been making insulin probably for about 30 years now, and so they've switched off of extracting it from animals so that you can feel good about using the insulin that you use instead of having to harvest it from a horse or from a rabbit. And so next we're going to talk about the structure and function of the genetic material. So genetics is the study of genes and how they carry information and how that information gets expressed, as well as how those genes are replicated. Chromosomes are the structures containing DNA that physically carry out hereditary information. The chromosomes contain genes. And genes are segments of DNA that are going to encode functional products, usually proteins. The genome happens to be the genetic information in a cell, all of it. And so the genetic code is a set of rules that determines how a nucleotide sequence is converted into an amino acid. And so the central dogma of modern biology is that DNA is transcribed into RNA and then translated into proteins. Now, I'll say that this is the language here in DNA of nucleic acids. and then RNA is also in nucleic acids, but proteins are in amino acids. And so there's the difference there. Now, I'll also mention one more thing that I mentioned previously, is that we have the DNA being able to self-replicate. And so this is, I'm going to put an S there. This reminds us that S means synthesis here. And so we're able to copy that DNA. So next we have these two terms, genotype and phenotype. And genotype is the genetic makeup of an organism, while the phenotype is the expression of the genes. And so the genotype is kind of like the blueprint, but the phenotype is kind of like what the actual house or building actually looks like. And so bacteria usually have a single circular chromosome that's made of DNA and associated proteins, and short tandem repeats are repeating sequences of non-coding DNA. And so here we can see the prokaryotic chromosome. It's coming out of the cell, and so you can see how some of them are very large and some of them are going to be smaller. And so this is just an example of one of them. So the flow of genetic information. Vertical gene transfer is the flow of genetic information from one generation to the next. So you guys probably have two parents. Well, all of you have two parents. So you have a mom and a dad who got together and decided to mix their genetic information, and that produced you. And so here you are with half of your genetic information from your father and half of your genetic information from your mother. And so you probably have a mixture of characteristics of them. In bacteria, we have some different things going on. So we can have the expression, so the genetic information is used within a cell to produce the proteins needed for the cell function. We can also have recombination, where it can be transferred horizontally between cells of the same generation. And so we'll talk about horizontal gene transfer, which is a little bit different here. And then we can also replicate it, like we can the same way that humans replicate in mitosis. So DNA replication is where DNA forms a double helix. Okay, this is the backbone that's going to consist of deoxyribose phosphate. Two strands of nucleotides are held together by hydrogen bonds between the A's and the T's and the C's and the G's. And these strands are what we call anti-parallel. And so the order of nitrogen-containing bases forms the genetic instructions of the organism. So here we can take a look at this. We can see what's going on here. And so we have the genes, and they're basically opposite of each other. And so the T's and the A's are always going to bond together, and the C's and the G's are always going to bond together. And that's how we're able to kind of make a copy of this, because it's semi-conservative. And so one thing I will point out to you is the number of dots between here. Okay, so the C's and the G's have 3 hydrogen bonds. Now, we all know that hydrogen bonds are very weak, and these weak hydrogen bonds are great. However, if you have more of them, it's going to be harder to break apart. So the A's and the T's have less kind of stickiness to each other than the C's and the G's. So one strand serves as the template for the production of the second strand in DNA replication. Topoisomerase and the gyrase relax the strands, helicase separates the strands, and the replication fork is created. And so this is that first step here. So the double helix of the parental DNA separates the weak hydrogen bonds between the nucleotides on the opposite strands, break in response to the action of the DNA replication enzymes. And so hydrogen bonds are formed between new complementary nucleotides, and each strand of the parental template is now going to form new base pairs. So we're going to have different ones coming in here. So they're going to come in here, and they're going to kind of insert themselves. So we got extra nucleotides floating around so that we can make this DNA. And so enzymes are going to catalyze the formation of the sugar phosphate bonds between the sequential nucleotides, resulting in each daughter strand. So DNA polymerase adds nucleotides to the growing strands. It always moves in the 5' to 3' direction. If you were in my classes for anatomy and physiology, or even biology, you probably heard me say, hey, it always moves in the 5' to 3' direction. Now, then I didn't care that you know that. Now I do care that you know that. And this is going to be initiated by an RNA primer, and the leading strand is going to be synthesized continuously. Okay? The lagging strand is going to be synthesized discontinuously, and it's going to create what we call Okazaki fragments. And these DNA polymerases remove the RNA primers. These Okazaki fragments are joined together by the DNA polymerases and the DNA ligases. And so I've just thrown a bunch of terms at you, and I understand that. And these are all of the enzymes that are used in DNA replication, expression, and repair that are going to be important that you need to know them. I would commit these to memory. So in figure 8.5, basically what's going on at the replication fork. And so these enzymes are going to unwind the parental double helix, and then the proteins are going to stabilize the unwound parental DNA. Now, because it's going to want to go back into its original shape. So then the DNA polymerase comes in, and like I said, it's going to be synthesized continuously. And so you can see how this is a continuous line here. And so now we're going to go over to number 4. The lagging strand is synthesized discontinuously. Primase and RNA polymerase synthesizes a short RNA primer, which is then extended by the DNA polymerase. And so we start with this RNA primer, and then the DNA polymerase comes in and helps it out. And so the DNA polymerase digests the RNA primer and replaces it with DNA. And then we have DNA ligase that's going to come in here and fill these breaks in these gaps, these Okazaki fragments. So basically what it does is it glues them together. And so energy for replication is supplied by the nucleotides. So the hydrolysis of the 2 phosphate groups on ATP provides that energy. And so here we can see that. So, whenever you have some type of nucleotide, you generally have the 3 phosphates attached to it. And so what they do in order to do this is by splitting off the first 2, so they make it an adenosine diphosphate and then an adenosine monophosphate, or whatever nucleotide it is. And that hydrolysis leaves these to go out, and they get reused later on. But then we put all this together. So DNA synthesis is a very energy-intensive process. Most of the stuff we're going to be talking about today is a very energy-intensive process. And so most bacterial DNA replication is bidirectional. So each offspring cell receives 1 copy of the DNA molecule, and replication is highly accurate due to the proofreading capability of DNA polymerase. And so here we can see this. What's going to happen is we create this replication fork here, and then you can see how it goes off, and then eventually we get that termination of replication, and we get 2 essentially equal cells, or equal genomes down here. So here's your check your understanding question. Make sure you guys understand this before moving on. So RNA and protein synthesis. Ribonucleic acids, these are going to be single-stranded with a 5-carbon ribose sugar, and it's going to contain uracil, U, instead of thymine. So ribosomal RNA is the integral part of ribosomes. Transfer RNA transports the amino acids during protein synthesis, and messenger RNA carries coded information from DNA to ribosomes. The synthesis of complementary mRNA strand from a DNA template is what we call transcription. And transcription begins when RNA polymerase binds to the promoter sequence on DNA. And it's always going to proceed in the 5' to 3' direction. Only one of the 2 DNA strands is transcribed. And so the transcription stops when it reaches the terminator sequence on DNA. So this here is the process of transcription. And so what's going to happen is the RNA polymerase is going to bind here, and it's going to start to make this mRNA transcript off of this. And so what it does is it makes it and it starts shifting it along until you kind of get this very long kind of stringy transcript. Now this is single-stranded RNA here, and you can actually see at the end it pops off. And so the translation is the second part of this. It's the mRNA is translated from the language of nucleic acids into the language of proteins, which is amino acids. And so codons are groups of 3 mRNA nucleotides that code for particular amino acids, and we have 61 sense codons that encode the 20 amino acids. And so the genetic code involves degeneracy, meaning that each amino acid is coded by several different codons. And so here you've probably seen this chart before, and so a couple of the ones that I'll point out to you. So this is the chart that tells you, in that particular codon where everything is going to be. And so the ones that I'll point out to you, and I'll point them out to you in the way that I want to. AUG down here is what we call start. Now, how do I remember AUG as start? Well, generally the school year starts in August. So that's how I remember that. Okay, Now, the other three that are really important are the stop codons. And so the stop codons are over here. This is UAA, UGA, and UAG. So UAA, UGA, and UAG are all stop codons. And so the stop codons, they actually cause the protein synthesis to stop. Now, how do I remember this? Well, you have UAA, and it's like, you know, if you're talking to your little sister or little brother, you are annoying, and then UGA, you go away, and UAG, you are gone. So translation of mRNA begins at the start codon AUG. It always begins there, and it ends, like I said, at the UAA, UAG, and UGA. And the codons from mRNA are read sequentially. So the tRNA molecules transport the required amino acids to the ribosomes, and the tRNA molecules have what we call an anticodon. So that's going to be the opposite of the of what we see on the codon. And so the amino acids are joined by these peptide bonds. All right, so here is the process of translation. And so we've got the ribosomes, and remember the ribosomes here, these are going to be made up of both RNA and some protein. So the ribosomes are right there. Those are the ribosomal subunits. You've got the small unit and the large subunit that are going to come together. The other things that I'll point out to you are the tRNA here, as well as the anticodon. And so the anticodon is going to match up with this. And so if you use the law of complementary base pairing, A is going to bond with U in RNA, U is going to bond with A in RNA, and so is G and C are still going to bond with each other in RNA. Now let's erase this just so we can have a little bit more cleanliness on here and we can kind of see what's going on. And so what we have going on is this anticodon here is going to come in here and it's going to do that. And it's gonna bind to that. And so that's going to bring the ribosome together. And so that's where we're going to go into this one. So on the assembled ribosome, the tRNA carrying the first amino acid is paired with the start codon of the mRNA. This is the place where the first tRNA is called to the P site and a tRNA carrying the second amino acid approaches. And so this one in particular is a leucine that's attached to it, and it's got AAU for the second codon being UUA. So next we have the peptide bond formation, which is a type of covalent bond here. And we've got three different sites here. And so I want to talk to you guys about what these sites are. And so this is the E site, this is the P site, and this is the A site. And so the E site is for exit, the P site is the peptidyl site, and the A site is the amino acyl, amino ACYL. And so what it's doing is it's the different types of bond that it's going to form. Now the next thing that happens is all of these things kind of start moving through the ribosome. Everything's kind of moving this way. And so eventually what's going to happen is in the E site here, the initial tRNA is going to get kicked off, and the other ones are going to start moving over, and so they translocate. And so then we've got a phenylalanine here coming in, and we're going to continue doing this code until we get to a stop codon. And so we're going to continue going on, we're going to continue lengthening this growing polypeptide chain here until we get to that stop codon. And so here we get to UGA, which is a stop codon, and this polypeptide gets released. What actually happens chemically to it is you get a water molecule added to the very end of it, and it separates from that growing polypeptide chain. And so finally the last tRNA gets released, the ribosome separates, and the released polypeptide forms that new protein. In translation, in bacteria, translation can begin before transcription is actually complete. So they can start actually making the proteins before they're done actually making the mRNA transcript. And so simultaneous transcription and translation in bacteria, basically what it does is it takes the RNA polymerase and it spits it straight into a ribosome, and that ribosome is going to start making the proteins coming out of it, which is really an interesting thing if you start to think about it. In eukaryotes, transcription occurs in the nucleus, whereas translation occurs in the cytoplasm. So a very good question might be to ask, hey, why can we have transcription and translation happening at the same time in the bacterial cell but not in eukaryotic cells? In eukaryotes, we have exons and introns, and exons are regions of DNA that code for proteins, whereas introns are regions in DNA that do not code for proteins. And so small nuclear riboproteins, or snRNPs, remove the introns and splice the exons together. And so here we can see this. So we've got these exons. We'll call this exon number 1. We'll call this exon number 2 and exon number 3. And so what happens is in the nucleus, the snRNPs are going to remove these introns, and so they're going to put them back together. Now, the other thing that they can do is they can actually rearrange all of these to have a different order. And so we started with 1, 2, and 3, but the order could end up being 3, 2, 1, or something different like that. And so this explains how we have, something like 20,000 genes, but we can make hundreds of thousands of proteins. So here's your check your understanding question. Please make sure you guys understand this. If you don't, please ask a question. So next we're going to talk about the regulation of bacterial gene expression. And so constitutive genes are expressed at a fixed rate, and in other genes they're expressed only when they're needed. And these are what we call inducible genes, repressible genes, or catabolite repression. Repression inhibits gene expression and decreases enzyme synthesis. It's mediated by repressors, proteins that are going to block the transcription. And the default position of a repressor gene is on. And so the induction turns on the gene expression. It's initiated by what we call an inducer, and the default position of an inducible gene is off. So the promoter is a segment of DNA where RNA polymerase initiates the transcription of structural genes. The operator is a segment of DNA that controls the transcription of structural genes, and the operon is a set of operator and promoter sites in the structural genes they control. An inducible operon is a and structural genes are not transcribed unless an inducer is present. In the absence of lactose, the repressor binds to the operator, preventing transcription. And in the presence of lactose, the lactose inducer binds to that repressor, and that repressor cannot bind to the operator, and transcription occurs. So this is the lac operon. This is one of the first ones they discovered. That's why they talked about lactose in there. And so here you can see this. So we've got the operon here. And so the structure of the operon consists of the promoter and the operator sites and the structural genes that code for the protein. The operon is regulated by the product of the regulatory gene I. And so the repressor, the active repressor, the operon, is off. And so the repressor protein binds with the operator, preventing the transcription from the operon. And so the repressor inactive, where the operon is on, when the inducer allolactose binds to the repressor protein, the inactivated repressor can no longer block transcription, and the structural genes are transcribed. This ultimately results in the production of enzymes needed for the lactose catabolism. And so in repressible operons, structural genes are transcribed until they're turned off. Excess tryptophan is a co-repressor that binds and activates the repressor to bind to the operator, stopping tryptophan synthesis. And so here is a repressible operon. And so this structure is very similar to the other one. And the operator sites are structural genes that code for the protein, where the operon is regulated by the product of the regulatory gene I. And so in this case, we have the repressor being inactive, and the transcription and translation proceed, leading to the synthesis of tryptophan. But when we have the tryptophan there and the repressor is active, the operon gets turned off, the co-repressor tryptophan binds to that repressor protein, and the activated repressor binds with the operator, preventing the transcription from the operon. So here's a check your understanding question. These things don't make sense when you first look at them, and that's okay. Sit down, write it out, and kind of make sense for it for yourself. It's very important that you're able to do this. So positive regulation can come from catabolite repression, which inhibits the cells from using carbon sources other than glucose. And cyclic AMP can also aid in, is going to build up in a cell when glucose is not available. And cyclic AMP binds to the lac promoter that initiates transcription and allows the cell to use lactose. So here, bacteria growing on glucose as the sole carbon source grows faster than those growing on lactose. And so bacteria growing in the second one in this medium containing glucose, and lactose first consume the glucose. Then, after a short lag time, the lactose begins, and so the cellular cyclic AMP increases, and the lac operon is transcribed. More lactose is transported into the cell, and beta-galactosidase is synthesized to break down the lactose. So the positive regulation of the lac operon starts with the lactose being present, the glucose is being scarce, which means the cyclic AMP level is high. So we've got a lot of the cyclic AMP, and we've got the inactive lac repressor. And so if glucose is scarce, the high level of cyclic AMP is going to activate the CAP, which is this protein that comes over here and promotes this. And so the lac operon then produces large amounts of mRNA for lactose digestion. And so so it's going to continue to break down that lactose. With the lactose present, the glucose is present, but the cyclic AMP level is low. When glucose is present, the cyclic AMP is very scarce, and the CAP protein is unable to stimulate the transcription. Okay, because it's going to have to bind with everything that it comes together with. And so epigenetic control. So methylating nucleotides turns genes off. You might have heard of epigenetics before because it's one of the faster growing segments of biology. So methylated genes can be passed to offspring cells. This is not permanent, but you have to be able to edit it somehow. And so microRNAs, or miRNAs, are going to base pair with mRNAs to make it double-stranded. And so this is a way of post-transcriptional control. And so the double-stranded RNA is enzymatically destroyed, preventing a production of a protein. And so here we can see this process. The transcription of mRNA occurs, but the miRNA, the microRNAs, bind to the target mRNAs that has at least 6 complementary bases, and it keeps it from actually being able to be transcribed. It actually degrades the mRNA. So here you check your understanding questions. Please make sure you understand them before you move on. So let's talk about how we get changes in the genetic materials. A mutation is a permanent change in the base sequence of DNA, and mutations can be neutral, beneficial, or they can be harmful. And mutagens are agents that are going to cause mutations. We also have spontaneous mutations that occur in the absence of a mutagen. And so base substitution, this is the change in one base of DNA, or what we also call a point mutation. And so what's happened here is during DNA replication, a thymine is incorporated opposite a guanine by mistake. And so if not corrected, the next round of replication, the adenine pairs with a new thymine, yielding an AT instead of a CG. And so when the mRNA is transcribed from the DNA, we get a different codon, or we could get a different codon. And so in this case, we would have, instead of cysteine and cysteine, we'd have a cysteine, then a tyrosine. A missense mutation is where the base substitution results in a change in an amino acid. And so here you can see the DNA template strand that's going to be transcribed into mRNA, and we get this normal DNA sequence here. So methionine, lysine, phenylalanine, and glycine, and then we get a stop. And so in a missense mutation, we get a methionine, a lysine, a phenylalanine, and a serine. And so what's happened here is this very last one. Instead of having a G and a C, we've had an A and a T. And so this is when we get that extra, that missense mutation. A nonsense mutation is a base substitution that results in a nonsense or a stop codon, and so it's going to actually break the protein. And so here we get a premature stop codon, so this lysine is that second protein, but instead of having an AAG, we get a UAG here on the mRNA. So instead of having this sequence, we get this sequence, and so then we get a UAG, which is a premature stop there. We can also have a frame shift mutation where we can either insert or delete one or more nucleotide pairs, and this shifts the translational reading frame. And so here we can see this. So what happens is we go from a methionine, lysine, phenylalanine, glycine, and then a stop. We get methionine, a lysine, then we go to a leucine, then an alanine. And so we've completely changed the protein, and it's going to be a completely different kind of setup than what we had previously. So here's your check your understanding question. If you don't understand, please make sure you ask a question. So chemical mutagens like nitrous acid are going to cause adenine to bind with cytosine instead of thymine. A nucleoside analog incorporates into DNA in place of a normal base and can cause mistakes in base pairing. And so this is what that is. That nitrous acid causes this. And so this is going to cause just some changes in that chemical structure. And so what this ends up doing is we get that HNO2 in there, and then it alters the parent DNA, and then we get replication. So one of them is going to get normal daughter DNA, while the other one is going to get altered DNA. And so then we have mutations and altered DNA in the granddaughter kind of generation. And so the normal nitrogenous bases are here on the left, but then the analogs are there on the right. And so those analogs react completely different. And so what they can do is, in the first one, they can the adenine can pair with, the 2-aminopurine is incorporated into the DNA in place of an adenine, but can pair with the cytosine. So the AT pair becomes a CG pair. And so in the bottom one, the 5-bromouracil is used as an anti-cancer drug because it is mistaken for thymine by cellular enzymes, but it pairs with cytosine. So in the next DNA replication, an AT becomes a GC. And so radiation can also cause mutations. So ionizing radiation, X-rays, and gamma rays causes the formation of ions that can oxidize nucleotides and break the deoxyribose phosphate backbone. And so UV radiation can also cause thymine dimers, and we're very familiar here in South Florida with these UV radiations. Photolysases, these are going to separate those thymine dimers, and nucleotide excision repair is where enzymes are going to cut out correct bases and fill in the correct bases. And so here we've got the ultraviolet light beating down, and the thymine dimers is going to cause them to actually bond to each other instead of bond to their complementary base pair. And so what happens is the endonuclease goes in and it cuts the DNA and adds the exonuclease, and it's going to remove that damaged DNA. The DNA polymerase then fills in the gap, synthesizing the new DNA using the intact strand, and the DNA ligase seals up the remaining gaps by joining the old and new DNA. If this doesn't work out, this is a very quick way for you to get skin cancer. This is also a very quick way for microbes to die. And so, one of the best ways to disinfect something is, putting it into UV light. And when you put something into UV light, it starts to break down the bacteria. And so the spontaneous mutation rates are about 1 in 10 to the 9 replicated base pairs, or 1 in 10 to the 6 replicated genes. And mutagens increase the mutation rate to 10 to the -5 or 10 to the -3 replicated genes. So here, as your check your understanding questions, please make sure you understand these. If you don't, please ask a question. So let's identify some mutants. So positive or direct selection directs mutant cells because they grow or appear different than the unmutated cells. Negative or indirect selection detects mutant cells that cannot grow or perform a certain function. Auxotrophs are mutants that have a nutritional requirement present in the parent. So this is like the use of replica plating. So replica plating, it's when you start with sterile velvet is pressed down on the colonies of a master plate, and that master plate with medium containing the histidine is then basically we're going to use them to push down on these other ones. So we transfer them to new plates, and so then the plates are incubated. So these petri dishes contain different mediums. So one has histidine, one does not have histidine. And so then you can kind of look and see which one is the mutant, based on if it's missing or not. And so this is one of those things where, you're looking for, those, what's it called? Those mutations that are beneficial, essentially. The Ames test exposes a mutant bacteria to mutagenic substances to measure the rate of reversal of the mutation. This indicates the degree to which a substance is mutagenic. And so this Ames test here, we get 2 cultures. We're prepared, in this case, salmonella bacteria that have lost the ability to synthesize histidine. And the suspected mutagen is added to the experimental sample only. And so the control is going to be normal. It's going to be the rat liver extract. But then, each sample is poured into the plate, and the medium lacking histidine, and the plates are then incubated at 37 degrees Celsius. And so then, once you put the mutagen in there for the experimental one, you'll see it grows a lot more than the control plate. So here are your check your understanding questions. Please make sure you take a moment to take a look at these. So let's talk about genetic transfer and recombination. So we talked about vertical gene transfer earlier. Now we're going to talk about horizontal gene transfer, which is where you can, where bacteria can actually transfer DNA from one another to each other in the same generation time. And so genetic recombination is a exchange of genes between two DNA molecules that creates genetic diversity. And crossing over is two chromosomes break and rejoin, resulting in the insertion of foreign DNA into the chromosome. And so crossing over is what we do, is what, why you kind of look like your mom and your dad. And so what happens is we get the Rec-A protein catalyzing the joining of the two strands, and then the recipient's chromosomes contain the new DNA, and the complementary base pairs are switched on the other one. And so the donor DNA will be destroyed. The recipient may now have one or more new genes. So vertical gene transfer, like I said, is from parent to offspring. But the horizontal gene transfer is genes between cells of the same generation, or even different generations, depending on the lifespan. So transformation is when genes are transferred from one bacterium to another as naked DNA. And so here's what's happening. So we've got the living encapsulated bacteria. So what they're doing here is they're demonstrating genetic transformation. And so this living bacteria is encapsulated in bacteria that's injected into the mouse. The mouse then died. Okay, so then they take the colonies of encapsulated bacteria were isolated from the dead mouse, and the living non-encapsulated bacteria were injected back into the mouse, and the mouse remained healthy. A few colonies of the non-encapsulated bacteria were isolated from the phagocytes and destroyed the non-encapsulated bacteria. Then they heat killed the encapsulated bacteria and then injected those into the mouse, where the mouse remained healthy and no colonies were isolated. And then lastly, the living encapsulated and heat-killed encapsulated bacteria were both injected into the mouse, and then the mouse died. And so the colonies encapsulated bacteria from this were all the dead mice. And so what's happening here is we're having that recombination. And so what's happening is when these cells die, they reach out and they grab this DNA. It's kind of what we call a funeral grab. And so the mechanism of genetic transformation in bacteria is when the recipient cell takes up the donor DNA, and that donor DNA aligns with complementary base pairs in the genome, and the recombination occurs between the donor DNA and the recipient DNA, and we have some degraded unrecombined DNA, and we get a genetically transformed cell. Conjugation is where plasmids are transferred from one bacterium to another, and this requires cell-to-cell contact via what we call sex pili. And so here you can see some pictures of that. So you see this very long kind of stringy thing. These things are in the process of trading DNA. And so you can also see one right there. So what happens is the donor carry the plasmid, the F factor, and are called the F positive cells. The HFR cells contain the F factor on the chromosome. And so we get this this bridge, the mating bridge there from that sex pili. And so the F positive cell and the F negative cell, so the F factor is present in the F positive cell, and it goes over there and it goes into the other one, gets copied, and then they both have that particular gene. And so that recombination then occurs and it gets integrated into the genome. And so the conjugation in E. coli, you get the replication, and then you get the transfer of part of the chromosome. And so in the recipient, the recombination of the HFR chromosome and the fragment of the F- chromosome. So conjugation can be used to map the location of genes on a chromosome. So this is a genetic map of the chromosome of E. coli, and so if you look here, this is where it starts, and it goes around this way because it's circular. And so what happens is we have all of these different different genes on this DNA that are important for the life of the E. coli. And so if without one of these, or with one of these, they're able to continue to live. And so what they might do is they might pick up a better or more efficient gene from a different E. coli. So transduction in bacteria is transferred from a donor cell to a recipient cell via bacteriophage. And you also have two different types of this. You have generalized and specialized transduction. And so random bacterial DNA is packaged inside a phage and transferred into the recipient cell. And so the specialized transduction is where the specific bacterial genes are packaged inside a phage and transferred to the recipient cell. And so here's what's happening. And so this phage is a virus, okay? So the phage protein coat is basically, it has some DNA inside of it, and so it's going to attach itself to the bacterial cell, and then it's going to invade the bacterial chromosome. It's then going to make a lot of its own copies, okay? So occasionally during the phage assembly, the pieces of the bacterial DNA are packaged into the phage capsid, and this donor cell lyses and releases the phage particles containing that bacterial DNA. Then that phage goes in again, and it can actually infect that host cell, and then you get the recombination occurring, producing a recombinant cell with a genotype different from both donor and recipient cells. And so they're actually thinking about using phage therapy as a last line of defense, and they've actually, against antimicrobial resistance, because bacteria can either be resistant to phages or they can be resistant to antibiotics, but you can't really have both. There's just not enough room for all of those genes in the genome. So here's your check your understanding questions. Please make sure you understand what's going on here. So plasmids are self-replicating circular pieces of DNA in 1 to 5% of the size of bacterial chromosomes. They often code for proteins that enhance pathogenicity of a bacterium. So the R factor, this is a type of plasmid. You've got the origin of replication, so there's a mercury resistance here, there's the sulfonamide resistance, streptomycin resistance, chloramphenicol resistance, tetracycline resistance, and then you've got the origin of the transfer over here, and then you've got the pillocin conjugation proteins. And so this R factor is very, very useful. In science, when you want to actually, make sure that your plasmid has been picked up, what you'll do is you'll put antibiotics into the plate that you're incubating, and you want to put those antibiotics in because you want to be able to quickly identify whether your plasmid was actually taken up or not. And so conjugated plasmids carry the genes for sex pili and the transfer of the plasmids. The dissimilation plasmids encode enzymes for the catabolism of unusual compounds, and the resistance factors encode antibiotic resistance. Transposons are segments of DNA that can move from one region of DNA to the other, and these contain insertion sequences that code for transposases that cuts and reseals the DNA. The complex transposons can carry other genes. And so here you've got the transposon, so that insertion sequence is the simplest transposon, contains genes for transposase, an enzyme that catalyzes transposition. And so this can jump around, this can move around. Transposons are basically jumping genes. And so in this kind of thing, the transposase cuts the DNA, leaving some sticky ends, and then those sticky ends of the transposon target DNA and anneal, which means they come back together, and they insert themselves into that plasmid. So here's your check your understanding questions. Please make sure you understand what's going on. So the last one we're going to talk about is evolution. And so mutations and recombination create cell diversity, and that diversity is the raw material for evolution. And so natural selection acts upon populations of organisms to ensure the survival of organisms fit for a particular environment. So natural selection means the environment favors the survival of some genotypes. And so from where does that diversity in genotypes come from?